Now let us talk about another fundamental process of cell. It is called the transcription. And during the central dogma of biology, as we know, let me draw the central dogma of biology again. So what is the central dogma of biology? As we are moving from DNA to RNA, and then finally this RNA to protein. So what we are making these proteins. And why we are making these proteins? Because proteins are actually everything to make a life. So we have DNA. The DNA contains all those information, and when the DNA is transcribed, we form RNA. So what do we mean by transcription? Transcription happens here. So transcription means the production of RNAs from DNA. And as we know, in a cell, in a typical cell, there are different varieties of RNA that can be present. So what are those RNAs? We normally have three different types of RNAs. One is called the mRNA or messenger RNA, and especially this messenger RNA is the most important RNA for this protein synthesis. Okay, because this mRNA work as a workbench from other RNAs or for other RNAs to work and to synthesize a protein, because this mRNA actually contains all those necessary information to make a protein, and this mRNA are generally coded via the DNAs via this transcription machinery. And we also have tRNAs or transfer RNAs. These tRNAs are the carrier molecule for amino acids, and what they do, they actually carry amino acids, attach them with each other, with themselves, and they bring them and they sit on this mRNA, and they finally attach those amino acids together via the peptide bond to make protein. Okay, so tRNA is also important, and tRNA is also produced via DNA. And we also have another type of RNA which is called the rRNA or the ribosomal RNA. So this ribosomal RNA or rRNA is the important ingredient to make ribosome, which is a protein factory. So So if we talk about the protein production, then you need a factory to produce that, and that is provided by this ribosome. So we have ribosomes in the cell. Remember, ribosomes are present in the rough endoplasmic reticulum. Actually, they are present in the endoplasmic reticulum. Thus, the endoplasmic reticulum is called the rough, because we have this rough structure due to the presence of these ribosomes. Okay, so mRNA, that means a messenger RNA which acts like a messenger. We have tRNA, transcription transfer RNAs, which actually transfers those amino acids to make protein together, and we have this rRNA, which is an important structural ingredient for making ribosomes. So what are ribosomes? Ribosomes is actually made up with rRNA and several different types of proteins. It's a stack of protein and RNA together. Okay, so in case of transcription, what we are looking at the production of RNA, mainly the messenger RNA from the DNA. Okay, so this is a normal view of how transcription process works, and what do you mean by transcription, and why transcription is important in prokaryote and eukaryotic both case. In this picture, what we have a prokaryotic cell. In this cell, as we know, prokaryotic cells are not having those nucleuses, so they do not have those complex structures. So what they have naked DNA or genetic material in their cytoplasm, and when the mRNA is transcribed from this DNA, and as soon as it is transcribed, the translation is going on almost simultaneously with this. So this is the natural process of going on those protein synthesis along with the transcription process. That is what is happening in case of prokaryotic cells. But in case of eukaryotic cell, what we are looking at, in eukaryotic cell, we have this nucleus. So the DNA material is embedded in the nucleus. It is stored in the nucleus, and after the production of mRNA, that mRNA has to travel from the nucleus into the cytoplasm, where we can find those ribosomes, because ribosomes are placed in the endoplasmic reticulum, and those endoplasmic reticulum are presented in the cytoplasm. So those mRNA which are produced and again are processed. So what do you mean by this processing? In case of prokaryotic cell, we do not find this processing, but for the translation purposes, for the protein synthesis purposes, we need to process the mRNA which is produced from the DNA of eukaryotic cell. Because why? Because as I have told you, that the DNA of eukaryotic cells are actually contained many junk DNAs which actually do not code for any protein. So it is just stay there maybe they have some evolutionary purposes, but normally they do not code for any protein. So we need processing to delete those portions out and to take only those portions which carry the code for making proteins, so carry the code for attaching amino acids through the tRNA system. So the mRNA comes into the cytoplasm, and then replication is going on. So this is what in case of eukaryote is going. So in case of eukaryote, we are actually doing some of the extra steps during this transcription, but overall scheme is more or less the same. So if we look at the big picture of all these systems of central dogma of biology, so what is going on in this case, we are making RNA, mRNA from the DNA, and DNA always make tRNA and rRNAs, then those rRNAs are attaching with other proteins to make ribosomes, and those ribosomes sit in the endoplasmic reticulum. And in case of prokaryotic cell, the ribosome is just scattered in the cellular matrix or the cell cytoplasm. So in this case, these are some enzymes which carry these tRNAs, and these are the tRNAs, which is a characteristic structure of this loop type form loop. This tRNA with this is the amino acid here. So this tRNA comes in, it will attach to it. This is an mRNA, the green colored big mRNA. It works like a workbench for protein synthesis, and we have this tRNAs which attach to them with different anticodons, and to with which they are attaching is called the codon of mRNA. So we have codon for mRNA, anticodon for tRNA. And they attach with themselves, and then finally attach their amino acids together to make a polypeptide. That's how overall system of central dogma is achieved. Now let's talk about this concept, is the asymmetrical transcription. So what do you mean by asymmetrical transcription? When you transcribe mRNA from DNA, we are actually talking about two different strands. We are talking about a template strand. So again, in this case, one strand has to act as a template strand. Remember, in case of DNA replication, I have told you the previous strand or the parental strand acts like a template strand, but in this case we also have this parental strand like DNA, or all those DNA strands. Those strands are working like template strands. So those DNA are wrapping around, so DNA opens up. So when DNA opens up, one strand of them helps to produce mRNA, but the other strand is not doing anything to produce mRNA. So that is important in this case, because one of those DNA strands are going to produce mRNA. So suppose this strand, suppose this is an A strand, this is a B strand, and this A strand is working like a template to produce mRNA. So we are producing mRNA. So this green color thing is an mRNA which is sequenced due to the complementarity of this A strand. So in this case, A strand is acting like a template strand, and the other strand which is not acting like a template strand is called the coding strand. So why it is called the coding strand? Because as we know, coding or the code is found in mRNA. So if we look at the mRNA, so for example, we have suppose this mRNA. This mRNA has those nucleotide sequences, and the mRNA is single stranded. It has those nucleotide sequences open together. We call them the code for proteins, and we have tRNAs which are having those anticodons which we actually bind with them. And what do you mean by codon? Codon is an assembly of 3 nucleotide sequences that denotes one amino acid. So 3 nucleotide sequences, 1 amino acid. That's the basic of making a protein. So 3 nucleotide sequence, 1 amino acid. So 3 nucleotide sequence, 1 amino acid. That is the final rule of coding. Okay. So in this case, we will discuss about this coding in next time in different chapters, but in this case what we are looking at this mRNAs, and these mRNAs actually having this strand. So this mRNA which is produced by making this A as a template strand is the coding strand. It's a code, or having a code. And this opposite strand, which is the B strand, is also having the same nucleotide sequence like the mRNA which is just being produced. So that is why this B strand is called, the B strand of the DNA is called the coding strand, because the sequence or the nucleotide structure of this B strand is exactly the similar like the newly formed mRNA. Okay, so that's why this is called the asymmetrical transcription. Now let's look at the different types of RNAs. We have talked about them. So the mRNA, which is a simple linear form of RNA which contains those codons. So the main thing about this mRNA, it contains the codon. If you look at the rRNA, we are talking about to produce ribosomes. So rRNAs are attaching with other types of proteins to make rRNAs, and if you talk about the tRNAs, the tRNA is just a structure like this loop. So tRNA has 3 different loops, and in the lower loop is called the anticodon loop, because this loop contains 3 different nucleotide sequences which will bind with this mRNA codons. That's why we call them the anticodons. And tRNA has an attachment site for amino acids. So tRNA are the carrier of the amino acids, so this is the attachment site for amino acids. So that's an important thing about tRNA. Now let's talk about RNA polymerase, which is an important key player to transcribe proteins. We have talked about the DNA polymerases in the process of DNA replication, but in the process of transcription, RNA polymerase is the main part. So it is also a really complex structure. It is it's a multiple subunit structure, so, or we call it a quaternary structure of this protein. So it has 2 alpha subunits, it has 1 beta subunit and 1 beta prime subunit, and it has a sigma factor. So what are these things are doing? This alpha subunit, this 2 alpha subunit helps to determine the DNA to be transcribed. So what the DNA to be transcribed is determined by the alpha 2 subunit. So there are 2 different subunits of alpha which are determining that, and you have 1 beta subunit which acts like a catalyst, or this is the actual enzymatic subunit for that purpose, which starts the polymerization reaction, because we have to go through the polymerization reaction. Remember those polymerization reaction, we need to have 3' hydroxyl, and we need to also have 5' phosphate to carry on 5' phosphate, and not only 1 phosphate, 3 phosphate groups or 2 phosphate groups to carry on those reactions. We generally have 3 phosphate because we are talking about NTPs. We are talking about NTPs. So not DNTPs in case, because we don't want deoxy ribonucleotide sequence. In this case we want ribonucleotide triphosphates. That's important. And another thing, this beta prime subunit are the binding site, or it acts like a binding and opens up the DNA. So if we're talking about or looking about the similarities between the the transcription and replication process, the DNA has to be zipped or opened up. Okay, so that's why that work was done in case of DNA replication via helicase, but in this case that work is also be done by this beta prime subunit. And we also have the sigma factor. So what is this sigma factor? Sigma factor recognizes the promoter region. What do you mean by this promoter region? Promoter region is actually a very important region which tells the cell, or signals the cell to start transcription. So it's a start. It gives a signal to start. It is normally not the start point, but it gives a raise to start signaling. Okay, so whenever actually this RNA polymerase bound to a DNA, so we have a DNA RNA polymerase bound to it, and when the DNA is actually when the RNA polymerase is actually scanning the DNA, so have different subunits which are bound together. When it is scanning towards from one direction to another direction, when it ends up with a promoter sequence here, it actually triggers the cell. It says the cell, yes, there's a time to sequence the DNA, we encountered the right DNA to be transcribed, and then overall process is going on, and this promoter recognition is facilitated by the sigma factor. So if we look at this overall structure of this RNA polymerase, we have this 2 alpha subunit here, we have beta, which is the polymerizing subunit, you have the beta dashed, which act as a binding and a helicase like action on opening of the DNA template, and you also have finally the sigma factor, because the sigma factor is really important to recognize the promoter sequence. Okay, so this is the structure, how this overall system is going on. And again in this case we also have this one entry site and one exit site. So one exit site, one entry site is going on. So DNA is entering from one side and exiting from another side, and in both those sides DNA is wrapped around or coiled, but in the middle of the active site of this RNA polymerase, it is opened up, and we have the formation of new mRNA templates using NTPs. Okay, so let's talk about a little bit about the promoter sequences. As I have told, promoter sequence is give the sensation or the signaling for starting the transcription process. So normally what we have in most of the cases of prokaryotic cells or eukaryotic cell, what we are having an universal promoter which we call the consensus sequence. So this consensus sequence is more likely or more or less the universal sequence in the -35 or -10 prime upstream. So what do you mean by the -35 -10 region? So not prime, actually -35 and -10 region. So what do you mean by this? So let's discuss this. So if we have a DNA, we have different long nucleotide sequences throughout from this place to this place. So if we make the direction A to B, but this is not a way of maintaining the direction. We have, a scientists have different ways to memorize which direction we are talking about. If we are talking about the linear DNA or the circular DNA, we must have talk about a particular place, because in case of RNA synthesis the nucleotide sequence matters, but in case of DNA replication, what we know that ultimately we have to form the exact copy of that DNA. So we do not bother about whether it starts and whether it stops and all these things, but in case of this transcription process, we really bother about all these things. That's why in this case scientists are naming a process or a start point from where the transcription begins. They call it the point 1 or 0 to 1. So we have a point 0 from where it begins the transcription, and through where the transcription begins, we call those points or those sequences numbers, or each nucleotide sequence will be numbered 1, 2, 3 and like this throughout the process, and we call this direction the downstream region. We call them the downstream region. So if you can find this term many times in your book, so don't confuse at all. We call it the down. It is nothing about the up and down, but it's about the downstream, means the down then the 0, that means the higher values, and what we have before that region, before the starting point of transcription, we also have nucleotides, and we call those nucleotides, we are marking those nucleotides negatively. So -1, -2, -3, we call them the upstream region. So we have this upstream region in this direction. So this is what we are talking about. So normally what we are talking about means by promoter, we are meaning about the -35 -10 region, which is actually is telling us that we are talking about the 35 and 10 nucleotide upstream from the transcription sequence. So we have and that is totally logical, because we have to have the promoter region some about upstream. So suppose here we have that promoter region. So we have this promoter region, and we have a consensus sequence, or particularly same type of sequence repeatedly in different prokaryotes during different species and all these things, but we can find this throughout the DNAs of this. So we have the prokaryotic cell, and we have different types of DNAs to transcribe, but in all those DNAs, most of the time these sequences are conserved. That's why we call them the consensus sequence. Okay, so in the -35 stream, we have a particular sequence called TT-GACA. For example, this is given here, so it's a sequence of TTGACA. So it's a kind of a similar. I'm not telling about this in all of those DNAs. This is similar, and this is similar 100%, but this is similar maximum times. Okay. In another case, in -10 region, we have another sequence which is which is much more popular than TT-GACA, and which is more conserved than TT-GACA. It's called the Tata sequence. We also call them the Tata box, because it's a box-like structure, so we call the Tata box. So TATAAT. So this is the normal sequence. So it's a Tata box, which you can find in look at different kinds of expressions. So we have the tryptophan operon system. If you have the lac operon, if we are making a rec A protein or make a bad protein. So in all those proteins, in case of ARA operon, tryptophan operon, lac operon, and also we are making or talking about those SOS response to produce rec A proteins. In all those cases, so those are different types of protein sequencing mRNAs, but you can find those in all those different variations of mRNAs. This consensus sequence is almost the same between them. So one in -35 region, and then is 10 region. Okay. and we have some spacers. Spacer means some nucleotide sequences which are different from each other, but we have a little bit of similarity in these two regions, and this kind of similarities, and this actually is telling us that in evolutionary history those polymerase actually designed from a same organism. So we have a common ancestor where we have this RNA polymerase, and that polymerase is actually evolving like the same, and it actually reads all those different variations of organisms like the same. Okay, so let's move on to the next slide. Okay, so here what you have in this case, you can look at the structure. So let me erase all this thing. Okay, now let's talk about, so suppose this is a DNA. So we have a DNA here, and the transcription means the making of RNA from DNA. So we have the DNA, and we have this promoter sequence. As we know, in -35 region and -10 region, we have the TT, Kaka, and Tata sequence, and these sequences are the recognition site for starting the transcription process, and from where the transcription begins, we call them the 0 region, or from the 1 region we are actually making proteins. From this 1 region we are actually making proteins, or that means those DNA which is sequenced from 1 region towards 1, 2, 3, like this positive side, we are actually talking about the structural gene. So what do you mean by the structural gene? Structural gene means a gene which makes proteins, which helps to code proteins. Actually genes are the coding region of protein, so this gene code for protein. So those genes who non-code for protein or is not coding for protein will be called the regulatory sequence. In this case, all these regions from this -35 region, -10 region, and we have this promoter side, we have the operator side, and all these sites what we are having is called the regulatory sequences. Okay, so in this case, in the regulatory sequence, we have the Tata box and the -35 region. So how the process begins, actually, that's a very important thing to begin the process, and how the process begins is via the addition of this or the assembly of all the subunits of RNA polymerase. So if you are talking about RNA polymerase, we are talking about two alpha subunits, one beta dash subunit, and one sigma factor. So here comes the RNA polymerase. RNA polymerase binds to the regulatory sequence, and when it binds to the regulatory sequence, it also attaches to the sigma factor. And what it does is actually scanning through down this region, actually. So downstream, it is scanning downstream, and whenever it attaches with this consensus sequence of the promoter region, so we call it the promoter. What do you mean by the promoter? In this case, we mean by the promoter is the region where the DNA polymerase bind, and from where it regulates the processing of mRNA from DNA. Okay, so in this case, when it finds that place of the promoter region, it stalls there, and the sigma factor releases, and as soon as the sigma factor releases, this polymerase starts to polymerizing. So the beta subunit of that polymerase will start to polymerize the sequences using NTPs. Okay, that's how the process works. So here we